This semester, talking about rocks and things last week, right? Well, today we're going to get a little bit more personal. We're going to start talking about your personal body and how they work, and cancer and all that good stuff. So, we have with us today Santanu Dasgupta. Yes, he's coming to us from what used to be called the UT Health Center. Now they call themselves the UT Health Northeast. And they have a similar history to Laterno in many ways, because they were also an Army hospital, as Laterno was an Army hospital before it became Laterno University. So in some ways we are sister institutions here, and so it's nice to have people from the UT Health Northeast to visit us today. And Dr. Santanu is someone who came from India. He got his bachelor's degree in India and his PhD in India. He came here to the States to get a postdoctoral degree in biology, was it?
Cancer biology.
Cancer biology. And he did that at Johns Hopkins. And from Johns Hopkins he came here to Texas, because it's nicer to live in Texas than it is on the East Coast. Yes? Oh, wow. Yeah, anybody here from the Baltimore area or any place like that? No? Okay. All right, so let's give Dr. Santanu a warm welcome here.
Thank you so much for this warm appreciation, and thank you, Gary, for this nice little introduction. So to introduce myself, my name is Santanu, and basically my background is on cancer. So I'm going to talk about you. I know we have different backgrounds, and mostly we're students, so I will try to present as simple as possible, considering the enormous complexity of the disease though. So today I'm going to talk about that my lab is interested in developing blood test, or it's called biomarker, so that we can detect cancer pretty early, so that we can offer better treatment. And for that, we all know that we want to understand the disease first, how the disease occurs, how a normal cell in our body transforms into a cancer cell. And if we can detect the disease at pretty early, probably we can offer a cure. So at UT, I work with breast cancer. So it is a deadly disease and very high, particularly in Texas. And if you look at the data, the second most common cause of cancer-associated morbidity in states is around 14% of all new cancer cases. And the number of cases are very high, only in US, including the death rate. And family history has been known to be a strong factor. Am I standing on the right place? You guys can see the thing? Okay. 70% are ductal carcinoma. It's a pathologic term. I'll discuss that with you in a minute. And there are so far 3 known markers called ER, PR, and heart immune. These are 3 genes within our cells by which we can identify a breast cancer cell and give the patient some drug. But the problem is, even if you give those drugs, they don't cure the patients. The disease comes back after a certain time. So we want to get something out for detecting the disease at early. And the molecular progressions. What do you mean by molecular progression? As I told you earlier, how a normal cell, through changes in the cellular genes, transform into a cancer cell, that we need to understand. If we don't, it's very difficult to treat the disease. So that's our first goal is. And based on our understanding, we want to develop a blood-based, lab-based test, using which whether we can detect the disease early, and we can monitor the progression of the disease to offer therapy from time to time. So this model shows you how a normal cell becomes cancerous. This is a model of normal cell, and this is a real representation of how the ductal cell, when they're normal, looks like. And if you look at that, when the transfer, it's called DCIS, meaning ductal in situ carcinoma. That means when a normal cell is changing into an abnormal cell. See, there is there is a lining around the cell which is intact, but here there is no lining of the cells. They're kind of distorted, disintegrated. That means at this point the cell has started becoming abnormal. Can we identify these abnormalities earlier? And we do not know what are the earlier drivers, the genes, that changes this cell from normal to malignant. So when it becomes very dangerous, cancer, see, the cells look very different and they are traveling to the connective tissue. So they can break organs, tissues, and go from one place to another, which is very scary. And we all want to stop them. So this is, in nutshell, what are the different types of BC we got, like it's called TNBC, 10% meaning the cancer cells which do not express either HER2, ERBB or TR. HER2 means those cancer cells which express this gene, and you can give a drug against HER2 if the cells express this gene. But the problem is many patients even after treatment come back with the disease. So then you can't treat anymore. So you got to do something. And there are other group of patients called luminal, which is non-HER2, and it is also very difficult to treat. So these are in the most frequently mutated genes in the different molecular subtypes. Meaning, so far how much we know about the genes that are being altered in the tumor type. So these are a list of genes. You can really look around SPIGKC, GATA. There are many genes, but the overwhelming thing about it is we have still keep finding many other genes that are involved in this process of tumor genesis, making it much more difficult to develop therapies. So this is some genetic tests available in the market for determining the progression of the disease, but it is, even with the test, you cannot say whether someone is going to have a recurrence or a recurrent tumor somewhere in the body. It's very difficult. So these are the drugs that are available for treatment of the patients against HER2. And as I mentioned, these drugs work for some time, but eventually the disease comes back. And so this is the marker so far known for the identification of the disease recurrence, the lymph node status, the tumor size of the tumor, the grade of the tumor, and 3 markers. Other than that, to date, there is no known marker so that we can know how the disease progresses, whether you can detect it early. So at this point, my research comes. So we work on a molecule called SS3GL2 or endophysin. So this is a gene located on human chromosome 9p22. So we found that this gene is often deleted within the cancer cell in the BC patient. So we want to know why this gene is being deleted from the cancer cell. Does it have to do anything with the disease development when the gene is lost from the cells? So we wanted to know what is going on with this gene. So this is basically the structure of the gene. It has an N-terminal bar domain in a C-terminal SST domain through which it interacts with a lot of proteins, and it is present in different, other than human, in different animals like mouse, C. elegans, and others. And this is the way how it works. So our cells have several microvesicles which are all the time going in and out. And this protein that I'm working with, SST-GL2, help those microvesicles to go in and get out of the cells. And also it also helps in receptor tyrosine kinase signaling, trafficking within the cells and out. So this is their normal function. Now you can imagine if the gene is lost from a cell, so they won't be able to perform the normal function, right? So what would happen if the normal function of a gene is absent from a cell? The cell can become abnormal and can perform abnormal function. That's what we want to know. If there is a functional loss of this gene, how the cancer cells behave. So that's the whole study all about. And the next part I will talk about how this information we can translate into a medicine. So the first phase, what we do in the lab, we have several breast cancer cell lines. So we have MCF7, some 149 MD, MB 230. What are these? These are cells which were taken from the real tumor of a human, and in the laboratory we culture them and we can grow them giving specific food. So we took these three cell lines from three different humans and we cultured them in a culture cell. And these cells, we know for sure that they do not have this gene inside, and they have very low level, undetectable level of this protein. So what we did artificially, we engineered a bacterial plasmid, and within the plasmid DNA, we encoded this gene, and then put back to the cells. And this gene has a GFP, like a green fluorescent protein. Why it is there? So that if it goes inside the cell, the gene goes inside the cell, we would be able to see a green light inside the cell. This I will be showing. So if we see a green light inside the cell, that means we are successful to introduce the gene inside the cell. So these are all cancer cells. Now we want to see if the normal gene is functioning within this cancer cell, what happens to these cells? Can we stop the growth of these cells? And what happens? So this is very exciting. So next, what we did, we checked the proliferation of the cells, meaning how the cells are growing under the culture. So we did a test, and these column are the cells where we have the empty vector, meaning we did not put the genes in. And these are the different cell lines, MCF7, 3 different cell lines. And here we did not put the gene SSTG, but in these cells we introduced the gene, and you see that there is a significant, huge difference in the number of the cells of their growth. So they're not proliferating very well when you have this gene. So that means some way this gene SSTG2 is stopping the growth of the cancer cell. Oh, wow, that is very encouraging. But still we are on the culture plate. So next time what we wanted to see their anchorage independent growth. What is the meaning of this case? So cancer cell has the ability to grow independently without any growth support. Like if you put a agar, soft agar in a plate and put a single cancer cell and give them some food, they will grow. And when they grow, they form big colonies. And this is a typical character of a cancer cell. And so we put the cells in culture plate with agar and wanted to see how they form the colonies. And you see when there is no SST-gel to gene, they form good big colonies. But when we have the genes, they are forming colonies, but they are forming very less. That means not many cancer cells have the ability to grow in the soft agar. That's what we want. So, and another property of a cancer cell is called invasion. That means they can, I have shown you the slide that from one place, a cancer cell can travel to another through the bloodstream. And that is when they acquire the ability to invade through our body. So we wanted to know that how these cells are invading here. So when we don't have the genes, that's control cells, see, plenty of cells invade through in a culture plate. So what we do, we put a mesh, nylon mesh with lots of pore inside, and put the cancer cell on the top, and put some food at the bottom. So there is no food at the top. So cancer cells want to survive. They want to get the food. In order to get the food at the bottom, they're trapped in the nylon mesh that we put in between. It's just like a tennis court. You have something in the middle, right? So a ball can stuck into the net. So you put a net, but the cancer cells, in order to go to get the food, they trap into the net. And at that time, we take the net out. And see, the cancer cells where there is no SSTG2, they're invading a lot. But when we have the gene, they're invading still, but they're a lot less. So that means somehow this gene, when we introduced artificially into the cells, they're able to stop their independent anchorage, independent growth, their proliferation ability, and their invasion. So that is something very encouraging for us. Now, this is all phenotypic finding, right? We do not know what are the molecular outcome. What are the genes that the SSTGL2 is talking to have these changes at the cellular level? So another experiment would be very exciting, that we found that these cells, when have this gene SSTGL2, they are producing a lot of superoxide. Superoxide is normally produced by the mitochondria, and when there is a lot of superoxide in the cells can die. So we are wondering what is going on. We put a gene inside a cancer cell which are very aggressive growing, and as soon as we put the gene, they are proliferating less, they are not invading that well, and they are producing a lot of superoxide which can kill the cells itself. Maybe as a reason we are seeing that the cells are not growing well. This gene is doing something interesting inside. So we wanted to see what are the molecules they are playing inside the cells, and can we figure it out? So we looked at several genes within these treated cells. First one is MFN2, which is a mitochondria fusion factor, which controls the fusion of mitochondria, which is a very important process for regulating mitochondrial homeostasis within the cell. And pink one is a mitochondrial serine kinase, that also a part of the mitochondrial balance. MTTFA is a factor which regulates the transcription of the mitochondrial genome. And we see when we have the gene, their expression is kind of bumping up. The cells producing more of this protein. But the question is why? Does it have to do anything with the altered growth of the cells? They're not growing that well. So then what we did, and these proteins are specifically present in the mitochondria. You don't get this protein in the cytoplasm. So another question from these experiments stands up. SSTGL2 is a cytoplasmic protein. They work in the cytoplasm. And these proteins are in the mitochondria. Then how they're talking to each other? So there must have been a process through which a cytoplasmic protein is signaling through in the mitochondria. So first question that stuck into our mind, is this protein going to the mitochondria and then interacting with this? So in order to understand that, what we did, we took these cultured cells and isolated intact mitochondria from the cells. We have a procedure that we can do that. So there are no cytosolic protein here. We isolated total mitochondria. From the mitochondria, we isolated their protein. So what we have now, mitochondrial protein. So with the mitochondrial protein, we run this experiment. And you can see that in the mitochondrial protein, SSTGL2 is there, along with others. So these proteins are expected to be present in the mitochondria. That's fine, but this is not. So that means this experiment provides the first clue that SSTGL2 protein that we introduced in the culture cells, when we grow them in the lab, they are somehow traveling to the mitochondria and talking to this protein and allowing the cells not to grow that well. So in a way, we are preventing the growth of the aggressive cancer cell, which is good. We also looked at the release of a protein called cytochrome 3 from the mitochondria. You know, when mitochondria regulates apoptosis of cell death, and when it regulates cell death, it signals to several proteins, and one of the major protein is cytochrome C, which stores in the mitochondria. But when cells are going to die, it is destined to die, the protein comes from mitochondria to the cytoplasm, and it is released from mitochondria. So if you can develop a test through which you can know that the proteins, how much protein is in the cytoplasm or mitochondria, you can know whether apoptosis or cell death is induced. So we measured that, and we see when the gene is within the cells, most of the cytochrome C is not in the mitochondria. They are less in the mitochondria. Most of them are traveling to the cytosine C. That is quite wonderful. Like that means somehow mitochondrial death pathway is triggered into the cells following the introduction of this gene into the cell. So we are sensing a mechanism now. So this gene we put into the cells, it goes to the mitochondria, talking to some mitochondrial protein and allowing and stopping the growth of the cells. So we also see, so these are in the cell that the number of mitochondria are increasing when we have the gene in the cell, and they're kind of fused. Why they're fusing? So there is a possibility that so in a normal cell, so the mitochondria has to make a normal balance. Like you don't want abnormal or mutated mitochondria or ill health mitochondria. You need healthy mitochondria within the cell. So there is a process of mitochondria fusion through which mitochondria maintain a healthy mitochondria. So maybe through mitochondrial fusion, the cells want to keep normal healthy mitochondria within the cells so that the cells can undergo apoptosis and the tumor growth can slow down. And this is another experiment through which we proved that SSTGL2 protein goes to mitochondria. So what we did, it's simple. We take some cells, and as I mentioned earlier, that wherever this gene go into the cells, you can track it by this GFP tracker. So this cell has SSTGL2 protein with the GFP. And then the same cell was stained with a mitochondria marker called mitotracker. Many of them studying biology may be familiar with this. So now we want to see if SSTGL2 is in the mitochondria, both the colors should merge, right? The green and red signal should merge, and you can see some yellow signal. And you can see there are a lot of yellow signals within the same cells. That means definitely SSTGL2 protein is traveling from cytoplasm to the mitochondria and cross-talking with several molecules. And by this experiment, we wanted to know that MFN2 is a protein. We found that when you introduce SSTGL2, MFN2 kind of going up. And we found out by in silico analysis, like computer analysis, that they have a region where they can interact with each other, bind to each other. We don't know where the exact site is, but this is an arbitrary, that it's a predictive through computation. We know there are some sites where they can bind with each other. So based on the data, we wanted to see, can we pull them down together within the cancer cells, where we put the genes and within the mitochondria? And the answer is yes. So what we did, we isolated a protein from the mitochondria and probed them with both the genes, SSTGL2 and MFN2. And in all cases, we found that in the pooled protein, you have both these proteins sitting together. That meaning there is a possibility that they interact with each other and they signal to each other in the mitochondria to stop cancer growth. So this is another experiment which shows that this SSTGL2 and MFN2 protein, they are located in the same place of the cells. And the green signal is for SSTGL2 and the red signal for MFN2. If they merge, then you can see they are merged in many places in the cell, meaning they are working together. So till now, what I told that we took some cancer cells and we put the SSTGL2 gene inside the cells and see that it stops cancer growth. Now think about if we do the opposite. Now if you take a normal cells, which are normal, they're not cancerous, and they have normal SSTGL2 functioning protein, and if you now do the opposite, delete the protein from the cells, what happens? Can they develop tumor or they become tumorigenic? So we are doing the reverse experiment. So what we did, these are a bunch of normal cells. This is MCF10S cells, which is a normal breast epithelial cells which do not develop into cancer, but they grow in a culture plate. So what we did, we knocked down the SSTGL2 gene using an siRNA targeted to that gene in particular, and we make sure that gene is deleted to 80 to 90%. So when you delete this gene, you can see the MFN2, pink one, and this protein, which were like upregulated when you put SSTGL2, they are going down. That proves a definite connection between this gene and the other protein. And look at that, the phenotypic effect. When you knock this gene out, the cells started proliferating again. They're growing, they're behaving like cancer again, which is quite amazing. And they're forming colonies. They become more aggressive, invasive, and they're not forming that amount of superoxide. So now we transform a normal cell towards cancerous by deleting this gene. So think about in reality, if a normal person at some point of the lifetime has lost these genes, so there is a possibility that those cells can transform into malignant cells. And at that point, if you can intervene that by tracking this gene, probably you can prevent the disease. That's what we want to do. But in order to do so, we want to understand the mechanism. At what time point a gene can get lost from anybody's body, and what could be the reason, like stress or many other things that life can cause. We don't know. So we take another cell line, which is a slow-growing cancer cell line, and knock down SSTGL2, and we see the same thing. They're not growing that well. No, they're proliferating faster, they're invading more, and they're not producing superoxide, and they're producing less fusion bodies. Same is for the other cells. They're not producing that mitochondrial fusion bodies, and the number of mitochondria is also reduced, which is kind of very striking. We are working currently in the lab to understand the mechanism, how the mitochondria is getting lesser in number. What is the mechanism? So So the experiment so far we did is in culture plate, right? It's called in vitro experiment. Many of the biology students would do it in culture cells in the lab. But we want to see how these culture cells now behave in animal, like which is a representative model of human. Do they behave in the same way? Do they grow faster? And what are the molecular changes they acquire when they grow in the animal system? So we have it's called a orthotopic model. So we have taken female mice, and we take these cultured cells, two groups of cells. One group of cells has this protein, and another group of cells do not have this protein. So we take like 10 mice each group, and in the mammary fat we injected these cells. And we waited for 6 weeks or 7 weeks to see how this tumor behaves. And the uniqueness of this model is, if you put these cells here, they can spontaneously travel through the bloodstream in the lung and liver. And we wanted to see if we have this gene here, can we stop traveling of the cells from here to the other part of the body? If we can, so that is something very significant, very important. So everybody is clear on that. We took those cells, we inject those cells into the mammary fat, and we waited for 6 weeks for the tumor to come. And we see after 6 weeks big tumors, and we cut them out from the mice. And you can see in the control group where we did not put the gene, the tumor is kind of very big. They form very big tumor on the mammary fat. But comparatively, when you have the SSTGL2 gene, they grow tumor, definitely, but they are much smaller in size. So it is not completely curing the disease, which is unexpected by knocking out a single gene, but it is at some point it is reducing the size of the tumor. So that is very encouraging. So next phase we wanted to see, does it stop the cancer cell to go to the lung and liver? So first we check at the lung, and what we do, we take the lung out from the mice, and we look at the tumor. You can see multiple tumors. See how potent the tumor cells are. We put them in the mammary fat, but the tumor travels to their lung without doing anything in 5 weeks. So the cancer cell already have the program in their mind. It is all programmed that they know how to travel from one place to another. So that is dangerous. So, but whenever we have the gene SSTGL in the cell, we have lung bed, very few. So it's very encouraging for us. We are delighted to see that. So we take this lung out and we slice them and look at the tumor cell. You can see many tumor cells in here. But if you take the lung from these mice where have the genes, there are very few met, very few cancer cells. So that means that these mice will definitely survive longer. And if you can translate them into human, its effect is huge. So next we look at the liver, and you can see many liver metastases, and you cut it out and you can see the tumor cells. How do you know we have tumor cells? We cut them out, slice them and stain them, and you can identify cancer cells within the liver. They destroy the liver, they go into the liver, they blast it out and they grow. And when they grow, they are like a car without a brake. They do not stop growing, they just grow. But here you can see there is no met in the liver. That is quite exciting. Next we cut the tumor. We wanted to see that whether the tumor that we injected in the mice have enormous amount of SSTGL2 protein. And we see that these cells producing a lot of SSTGL2 protein and another protein called E-cadherin, which controls the metastasis. So if you lose this protein, cells can metastasis. So there is a loss of cell-cell contact. So this protein acts on the cell-cell contact. See, the cancer cell do not have a high amount of this. But when you introduce this gene, this protein goes up, coming back again. That means we can establish a cell-cell contact. When there is a cell-cell contact, the cancer cell grows slow. They cannot grow that fast. But a cancer cell migrate faster when they lost the cell-cell contact. There is no adherence between the cells. They change their shape, they travel through the bloodstream and go anywhere they want. So this is the most important part now I am heading to. So far the data I have shown, you might ask, oh, okay, all in cell culture, all in culture. What about in human? What is the expression level of this gene in human? Do we know that? No, we don't. So in order to know that, what we did, we have taken real tumor samples from human patients, 55 of them. And all of them have normal, their primary tumor and the metastatic tumor from the lymph node. So we collected their lymph node, the primary tumor tissue and the normal tissue from the same patient, and the number of total patients are 55. And we stain them, it's called immunohistochemistry, we stain them with an antibody specific to SSTGL2. And you can see this is normal. See, normal expression, the brown staining, you can see very deep, meaning the protein is expressing is a very high level in the normal. But if you go to their primary tumor, these are the tumor cells, and the tumor cells in the lymph nodes, they don't express, very low level. That means that this is a kind of protein which is sequentially being lost while a tumor is developed. And if we can monitor the loss of the protein, can it help us to detect the cancer early? That's the whole point of the whole study, whole talk. And we also see that MFN2, the another protein that we found that SSTGL2 is signaling to in the mitochondria, is also expressed very high level in the normal cells. This is normal tissue from the same patient. But if you go to their tumor, their expression is kind of very low and lost. So that means this is kind of, and many of the patients have this loss of this protein. So at this point, we can say that SSTGL2 is sequentially lost in cancer patients. And when we introduce this gene artificially into a cancer cell, it is stopping cancer cell growth. So that means, does it have any potential to bring it into the clinic? Can we develop SSTGL2 or MFM2 as a marker to get it to the doctors so that the cancer can be detected earlier? So, but in reality, the thing is, we have only 55 patients. The next step, what we need to do is to screen this molecule in hundreds of patients, like 500, 1,000, not in one lab. We might have to collaborate with many others around the world and within the states, and send them the antibody, and they can test it in hundreds of thousands of samples. If it turns out that this gene is really lost, and we can detect their prognosis or early detection, then it can be in clinic. That's what we want to do. So we also get tissue from normal humans who do not have any cancer. And we also make sure this protein is very highly expressed in normal tissues. And they are not from cancer patients, they are from normal individuals. To make sure what we are saying about the human tissue, it is correct. And in the human tissue, we also make sure that SSTGL2 is expressed in the mitochondria. What we did, we take this tissue, isolated the total mitochondria, and then from total mitochondria we make protein. So there is no contamination. And we make sure that SSTGL2 protein is in the mitochondria of the human tissue. So this is the first proof. So our study is the first study to show that SSTGL2 regulates tumor breast cancer growth, and they go to the mitochondria. And the most important slide of the whole talk is this one. Does it predict the disease progression? And it does. So when we compare using statistics, whether loss of SSTGL2 and MFN2 predicts poor outcome, and we can see that it does. So if you have a loss of SSTGL2 and MFN2 protein alone or in combination, they can kind of predict, okay, this patient is going to have recurrence or not going to respond to therapy. But still, it is a very low number of patients. We are planning to do in hundreds and thousands of samples the screening of these things. So we develop a kind of mechanism how the system is working. So what we believe is that it's a hypothetical cartoon. We believe that when we introduce SSTGL2 into the cells, in the cytoplasm, it is getting activated. We did not show you the data. And it now enters into the mitochondria and crosstalk with this protein and helping the mitochondria to become healthy, and they release a lot of superoxide to kill the cells by releasing cytochrome C, and as a result the cells are dying and tumors are getting shrink. And I think I would summarize and skip the next part of the talk. So to summarize, SSTGL2 is a potential tumor suppressor and associated with mitochondrial alterations. They frequently lost along with MFN2, and the loss of SSTGL2 predict poor outcome. This is the most important finding. And I did not show you the data. We also check the circulating exosome from this patient, and we show that normal exosomes from the patient carry both protein, but the tumors not. So what is the take-home message from here? That if we can monitor the loss of this protein in hundreds of patients, maybe we can develop someday a test by which we can detect cancer early and maybe offer a better cure. So I'll skip that part and go to that. So another important part is give me a second. Did we publish the data? So we have been able to publish the data in a good journal called Clinical Cancer Research, which is the American Association of Cancer Research Journal. And we published it, I think, last February. So it has been accepted, and we are going to the next phase of the study. So this is, I want to acknowledge a lot of people from the institutions. Dr. Stephen Adler, who is our dean of the institution, and provided the initial startup funding from the state. And Dr. Philly, Dr. Griffith working in my group as a collaborator. Dr. Wills is our pathologist. So what he does, he gives us all the human tissues from the hospital. So I have to go to the hospital, develop a IRB protocol, because he can't go to the hospital and say that, hey, give me some tissue. You can't do that. So you have to have a protocol, you have to have a pathologist in your team. So Dr. Wills works as a pathologist. So I am a member of the tumor board also over there. So I know what kind of patients are coming, and I get samples from him. Dr. Singh is our statistician. So whatever biostatistical program we ran through him. And Dr. Samprin is also a collaborator. Dr. Rikiv is our chair. We all do all the microscopy on the imaging with him. And Dr. Kandan sitting with me, he did all the work. And I would like to thank you for your patient hearing. And if you have any questions, please take it.
Lots of time for questions. So genetics students, biology students, now's your time. Questions from the audience.
Yes, please.
If we were interested in getting involved with your research, as far as internships or things like that, or just following with it, what would be the best way?
Pardon?
If we were interested in following your research as you guys continue forward, or getting in as far as internships or things like that, would we just contact the Science Center?
Yeah, you contact the Science Center, and we have a master's program, and you can get into the enroll. And the beauty of the master's program is you can be in a lab for 2 years, and you can get all the studies done. You will be given a project, and you can learn all the things, all the techniques, and you can write your thesis, and you can publish paper as an author.
Any summer opportunities for undergraduates in your lab?
Yes, there is.
There is.
Absolutely, absolutely. Undergrads are always welcome in the summer.
Okay.
You take a lot of students. Feel free to come, no problem. Absolutely, we provide lab support. You can learn some cutting edge techniques from the lab, and we can train you on the things. Pretty easy. Yes.
Is there any evidence of the protein that you were mentioning in patients that aren't cancerous? Is it possible that you can lose that protein and then not develop cancer in the future? Or
That's a great question. I like that. Yeah, but the problem is in cancer, there are two groups of genes we call. One we call is oncogenes, which are abundantly expressed in the cells, and you can target them. But cancer primarily developed by the loss of tumor suppressor that I talked about. Yeah, but in many cases, cancer cell not only loses one or two tumor suppressor, they loses a lot more, like 20, 30, 50, 100. So, yeah, losing one or two might not develop, but there is a possibility of the cells to become transferred if you have a loss of gene. Definitely, yes.
so it is an indicator, but
Yes,
not
yes.
necessarily.
So at that point, maybe if someone loses SST-GIL2 gene from a cells and multiple cells, it might take 10 or 20 years for the cells to develop malignant, along with other changes in the gene. But there is a hit there. It's sitting there, right? So that's the way cancer develops. Somebody is getting many of them are like sitting with some mutations. You need more. For example, colon cancer that we know, that was discovered in Johns Hopkins University, a very renowned scientist named Bart Vogelstein, which we call him like a god of cancer science. He discovered that there is a gene called APC, adenomatous polyposis coli. So there are 2 copies of each gene, right? So if you have 1 copy of the gene, polyp develops, right? So that's why the colonoscopy method has been developed. So we always look for APC gene mutation. If there is a biallelic mutation, in 100% cases, the person would develop cancer. So when we see a polyp with APC1 mutation, we cut those polyp out. So that's the way you can prevent cancer, by looking at a single gene. Is that powerful?
Other questions?
Yes.
So I know your goal is detection, but is there any possibility that some form of gene therapy would be able to help at least slow down?
That's a
cancer?
great point. But the problem with gene therapy, how would you be able to target a particular gene to specifically cancer cells? They're traveling everywhere.
Right. So does this gene cause damage to normal cells?
No, it doesn't. It
Doesn't.
doesn't. That's the beauty. So what we are thinking of, we are trying to use exosome to load this protein and give them to the cells. If it doesn't do anything, it will not cause any harm.
Right.
But some way you have to regulate the trafficking of the exosome so that they get into the tumor, right? So we have to put some marker or something. We are thinking many things out there. That's a good point. But gene therapy worked, but in many cases gene therapy failed because but whenever you put a gene in, tumor kind of develop a different memory. They can sense that you put something in, and they develop something else to get rid of the disease. For example, if you give for colon cancer, there is a common mutation called KRAS. So people have developed KRAS inhibitor, right? So you give an inhibitor to the colon cancer patient. So inhibitor is working for a few hours. After that, it doesn't work. So we found out that the tumor cells are developed another mutation in the same gene, and that the drug cannot bind anymore in the same region. They can even sense the drug binding pocket. So you develop a drug, put a drug, they can sense in 10 minutes, just like a virus or a bacteria mutate in a way. Cancer cell mutates so great, and they can develop another mutation and get rid of the drugs, and it doesn't work.
Not fair.
Yeah. So I don't know, within a short time frame, how a cancer cell develops such memory and backup plans is amazing, overwhelming.
Otherwise, I'm sure Dr. Santanu will stick around for a few
Yeah,
minutes to answer
sure.
more questions.
Absolutely.
And I have his contact information if you want to contact him. That's summer.
Absolutely.
Let's thank. See you all next week.
Yeah, and I made sure that they don't have to run for classes. Right?
Plenty of time. We're good.
Thank you all.